let me begin my talk with this. So I belong to Alberta Diabetes Institute at the University of Alberta. So this is the image that you see of the institute. Um, this institute is very unique and of its kind because it's centered for diabetes research. At the sixth floor of this is fifth floor of this is a human pluripotent stem cell facility that I have developed and initiated for the last 5 years. Um, it has also come in association with the clinical islet transplant program, which means that we transplant patients living with diabetes with cadaveric donor islets, and I will explain you in the later how what does that mean. On the seventh floor of this building, we have a GMP facility for cell therapy manufacturing. So it's purely a translational institution. But my journey never really begins with learning with stem cells for my degree in MSc or BSc or whatever. I really learned it and became a scientist when I was a young kid. And so when I was a very young kid, my mother actually planted a tree of a papaya tree in our backyard. Over the 2 years, this tree became grown up, became a large tree, a bigger tree, but and it started flowering, but it really didn't give. So I was always inquisitive to know why this tree is not giving any fruits, particularly it is so big. So I, at the same time, about principles of stem cell biology, tissue culture.
अच्छा, ये लैग हो रहा है क्या या मुझे ही सुनाई दे रहा है ऐसा समटाइम्स? हेलो पूनम मैडम?
नहीं सर, बराबर तो लग रहा है।
अच्छा, इसका मतलब इधर का इंटरनेट का कुछ प्रॉब्लम है क्या? बीच में जैसे लग रहा है कि आवाज चली जा रही है, ऐसा नहीं है? यस,
ऐसा?
हाँ,
Yeah, let me know अगर ऐसा कुछ होता है, so let me know. I can repeat it again, but I believe it's my internet is pretty
वो
good.
हो जाता है, मतलब बीच में चला जाता है।
No problem. I'll continue, but let me know if that happens. Please feel free to interrupt in between. Okay. Yeah. So when I was a very young kid, I started learning about plant tissue culture and stem cells, and also started learning the role of stem cells in plants. So I had an idea that what if I use these stem cells and transplant in this tree and graft it so that I should be able to transform this plant into a flowering tree. So what I did is basically I went to my neighbor's house and I got another tree which is a flowering tree and also fruiting at the same time. So the same papaya tree I took and I took the graft, a very young graft, which was a budding graft, probably has a stem cells in it. I took that out and I then grafted into my own tree in my backyard and transplanted the stem cells into a non-flowering plant. After a couple weeks or months, I was surprised to see that my plant started producing fruits, and it actually produced very delicious fruit for over 4 or 5 years. So that was my first experiment to learn about stem cell biology and the promise that stem cell regenerative medicine could do. So this is a very live and unique example, a true example which is associated with my own life. But since then, I began my journey in regenerative medicine, and I tried to explore what are the various possible alternatives for treating degenerative disease. As you can see over here, there could be many, many arms over here. It could be transplantation of cells or scaffolds, where you have gene therapy, you can make organoids, or maybe use stem cells to replenish older cells. But before we begin to that, let's understand the past present of medicine, how medicine actually originated, and what is the present form of medicine, and how what's going to be the future of it. So the past era of medicine is more intuitive medicine, which means that it is based on assumptions, it is based on observations, and more based on science and understanding the signs and symptoms. In past, the medicine and the history of medicine particularly is very old, and the most primitive one of that is Ayurveda, which is about 5,000 BC years old, right from the origination of Brahma to the late foundation of sculptures called Vakbhata. This is based on the 5 element of the Panchatattva you all know, and there are about 1,270 species of plants that has been introduced in this Ayurveda Granth, which probably counts about 8,000 or more than 8,000 herbal formulations. So this is the basis of India and Ayurveda. We have been getting medicines from since the last 500,000 years ago. There another forms of medicines came from Egyptian medicine, Chinese medicine, and others, which basically focused on utility of metals, needles, threads, and oils from Egyptian medicine. The Chinese medicine more like followed Indian culture as well, but they also introduced a newer concepts of acupuncture, acupressure, moxibustion, cupping, herbal medicines, and massage and exercise. Similarly, the Babylonian medicine focused on healing goddesses and using bandages and creams and pills for the diagnosis and progression of examination of diseases. The Greek and Roman medicines are fairly very young, and they basically focused on the supernatural powers, the hypocrisy, and also more onto the preventive measures, like prevent the diseases so that you don't get really infected. So the prevention is better than the cure, right? They focused on that. So that was the past era. Between 17th to 20th century, the form of medicines has really evolved, and we started learning about cells as the building blocks, and this allows studying the biology of the cells and tissues. Then we also introduced microscopy, so that we can started observing the life, the structure of the life, the ultrastructures for the disease and pathology. We introduced surgery to really understand the mechanism of treating injuries and repairs of tissues and damaged tissues. Then the understanding the pathology and mechanism of diseases that helps in correlating and finding from death to life. So this is all about 17th to 18th century, but in 19th and 20th century, the medicines evolved and more focused on the germ theory, the microorganisms that's growing, what is the diagnosis and technology that can be utilized for diagnosis of those microorganisms, radiology and microscopy and ultramicroscopy such as MRI, CT scans, and all this. And then finally, in 20th centuries, or nearly by the end of 20th centuries, we lived in the era for finding antibiotics. So in 21st century, that's where the concept of cell therapy or stem cell research has really introduced. So stem cell therapy is not very new. It actually first introduced as a concept in 1908, where the term stem cell was coined. And it's really between 1961 to 63, the first blood stem cell was discovered by Canadian scientist Tim and McCulloch. And since then, numerous scientists have discovered the concepts of stem cells in different areas of medicine, including the cloning of from a sheep cell by nuclear transfer, to introduction of induced pluripotent stem cells. Naka, a Nobel laureate who has discovered the discovery of induced pluripotent stem cells, and now this has gone far beyond to treating various diseases such as macular degenerative disease and eye disease, as well as heart failure and others. So there is a strong evidence for these stem cell utility in cancers and immune system, but we still have a wide range of disease that has not been worked out or well studied, particularly the heart disease, the Parkinson's, neurological disease, spinal cord injury, diabetes is one of them. And so there's a lot of work that has been undergoing in last 5 to 8 years, focused on these disease areas, and then several clinical trials have been initiated. So let's talk about what are stem cells, like, how do you identify these stem cells? Stem cells are particularly uncommitted mother cells with a miraculous capacity to indefinitely multiply and develop into any cell type of your body to form tissues and organs. Well, there are several types of stem cells. for example, totipotent stem cells, multipotent stem cells, pluripotent stem cells, oligopotent, and unipotent. And what is the difference between these stem cells? So totipotent stem cells are the one that differentiates into any cell type of the body, but they can also develop the extra embryonic tissues such as placenta and zygote. Pluripotent stem cells, on the other hand, they can only differentiate into the trigerm layers, which is ectoderm, mesoderm, and dendroderm. And these are the germ layers which give rise to the every single type of our body. But they cannot differentiate or develop into extra embryonic tissues such as placenta, cord blood cells, or zygote. Multipotent stem cells, on the other hand, are the cells that give rise to a very specific lineage. For example, if a cell has to go to make a blood cell, then these multipotent stem cells can only make different types of blood cells, but they cannot make skin cells or a lung cell, right? Same as oligopotent cells. Oligopotent stem cells are the one that differentiate into just one or two specific cell types within one or two cycle, proliferative cycle, and they ended up becoming more terminal cells. So for example, we have a blood mother cell, but this blood mother cell can only give to either B cell or T cell, but they are interchangeable, but it cannot make macrophages or NK cells or any other RBCs, for example. So that they cannot make this. Those are oligopotent stem cells. And then you have finally very committed lineage cells that are called unipotent stem cells, which means they can differentiate and develop onto one type of cell, such as epidermal scale cells or lung cell, but they cannot cross the barrier. So this is the hierarchy of stem cell gradation, and majority of the time we can utilize either pluripotent or multipotent stem cells for regenerative therapies, but oligopotent or unipotent has a very limited potential for repair and regeneration. So in order to in order to leverage the potential for pluripotent stem cells or multipotent stem cells, pluripotent stem cells are of two kinds. Either you have embryonic stem cell, or you can have an induced pluripotent stem cell. So induced pluripotent stem cells is relatively a newer concept with the discovery of Yamanaka factors from Shinya Yamanaka in Japan. And he basically introduced that there are a couple reprogramming factors that's called Yamanaka factors. These are basically the genes or the proteins that if you can do a forced expression into any adult somatic cell, that can basically rewind the cell back in time to become pluripotent like cells, and that's called induced pluripotent cells. So because you're inducing the stemness in the somatic cell, that's why they call induced pluripotent stem cells. Whereas embryonic stem cells are basically isolated from an inner cell mass of a blastocyst after fertilization. We can use both of these cells to basically generate all of the underlying cell types, including the progenitors as well as the differentiated cell types. However, the multipotent stem cells can only give rise to a lineage trajectory cell types. For example, ectoderm can only give rise to pigments and neurons and skins, but they cannot give to muscle cells or endothelial, and vice versa. Muscle and mesodermal can only give to muscle, endothelial, and MSCs, but not to skin or lung cell. And same as endodermal cells can give rise to only endoderm specific cells such as lung, pancreatic, and thyroid cells. So this is how the bifurcation and their function of stem cell occurs in nature. So if we talk about the promise of stem cell, the pluripotent stem cells are the one that has been shown a great promise and for therapeutic purpose. So what we get, how we can get that is if we can get a patient cell, such as a blood cell, we can reprogram these cells and become an iPS-like cell from the patient-derived cells. And we can now have an ability to generate a beating cardiomyocytes from these patient cells, such as a heart cell, which has a cardiac function, and they can actually pump blood circulation if they are transplanted into an injured heart. Similarly, these cells can also derive firing neurons, which have a very unique and specific electrical conductivity to generate a neuronal cells for regain the neuron function after a damaged nerves. Alternatively, we can also generate a pancreatic islet-like cells which can secrete insulin in response to glucose and probably help reversing diabetes, as what we are trying to focus in our own facility. So the utility of pluripotent stem cells have been explored for several years, and not only for the therapeutic purpose, but iPS cell applications can be to a diverse range, including the cell therapy, the disease modeling, drug discovery and development of newer drugs, personalized medicine, and testing and toxicology studies using iPS as a model system. And there are several companies around the world have used stem cell therapies for regenerative medicine, either as an autologous cell therapy, which means patient's own cell, your own cell is used and transplanted into your own body. That's called autologous. And an allogenic cell therapy means someone else's stem cells taken, expanded, and implanted into your own body. So this is foreign to your body, but they're allogenic, right? So all of these areas have been widely explored. And this is an evidence what I'm trying to show you here, this that several clinical trials have been initiated into several disease area, including bone, heart, cartilage, neuro, immuno, diabetes, lung, liver, gastrointestinal, and others. And as an example, there are two independent groups, one in the United States from Kapil Bharti and one in India in STEM Bangalore from Rajshri Paul. They have been successfully able to generate iPSC-derived retinal epithelial cells for the treatment of dry macular disease. And these two studies are in clinical trial, and cells have been transplanted in patients, and they have seen a very positive outcome for the regain of eyesight after dry AMD disease. In parallel, in the United States and in many other places around the world, iPSC-derived cardiomyocytes have been generated by Deepak Srivastava's group in Gladstone Institute, US. And they have been transplanting these cardiomyocytes generated from patient own cells in damaged heart to see the effect and regain the cardiac output and function for preventing heart diseases. Very similarly, iPS-derived neuronal cells have also been used for a treatment of ALS and Parkinson's disease. And two companies, Blue Rock Therapeutics and Kyoto University in Japan, they have initiated clinical trials generating neuron cells. So these are the three specific areas where the clinical trials have already promised. But there's another fourth area, which is diabetes, and that's where also the clinical trials have happened. And I'm going to talk about that in the later half of this. So two companies. First one was Vertex, that they have started a clinical trial with the induced pluripotent stem cell derived islet cell product that has been transplanted into 7 patients so far now, and 3 of them have been completely reversed with diabetes. This product is called VX880, and it requires full dose of immune suppression too. But basically, the concept shows that stem cell-derived islet completely reverse diabetes in these patients. And then similarly, 2 independent studies came from China very recently, where a 25 years old woman in China became the first patient who's been completely free and treated with type 1 diabetes using stem cell therapy. Here, they have used the woman's own stem cells and generate an islet-like, functional islet-like cells that secrete insulin and transplant it into the same patient, clearly indicating the promise of stem cells for treating both type 1 and type 2 diabetes. Now, why diabetes? Diabetes is really a global problem. We have about 828 million adults suffering from diabetes, and India is one of the second largest country with diabetes, patients in the world after China, and basically India is surpassing China very soon. But more importantly, only half of these patient population has received treatment so far, and other half is still diabetic. They're diabetic, and they really never received a treatment. So it's really an alarming situation, not only for the entire world, but specifically for India too, that we are slowly and not slowly, but rapidly and gradually progressing to diabetes and having diabetes in our subsequent generations for a long time. So what diabetes is basically, this is the picture you see a diagram of a healthy donor islet, which means that I have alpha cell, beta cell in blue, alpha cell in red, and other cell types. And the blue cells, which are the beta cells, are the ones that secrete insulin and regulate blood glucose. But if somehow these blue cells die, and if we can lose them, then it creates a progression of diabetes and basically creates a condition called type 1 diabetes if these cells are attacked by immune cells. If these cells are attacked by more kind of a stress or dysfunction, that means that the cells are still there, but they're unable to function due to the stress and environmental toxicants, then it's type 2 diabetes. And so what is the solution? In our own institution and University of Alberta, we have a long history of 25 years for stem cell-derived islet transplantation, as well as cadaveric donor islet transplantation. So my mentor, James Shapiro, Dr. Shapiro has developed the process called cadaveric donor islet transplant 25 years ago, which means that we harvest a pancreas from a deceased donor who died because of an injury or accident or something. We can isolate islets from the donor pancreas and then transplant those islets back into the liver of a recipient and a matched recipient. And this process is called allogenic cell transplant. But if we can isolate the islets from the pancreas from the same donor and transplant back into the liver intraportal system of the same patient, then it's an autologous cell therapy. And certain patients, such as with pancreatitis or pancreatic cancer, this therapy is very useful because the pancreas is very harmful and because of the cancer condition, but the islets are still functional. So because of the cancer, they have to remove the entire pancreas, so the patient becomes diabetic. But these cells, islet cells, can be transplanted back into the liver system, so they can be treated with by diabetes, but they are also free from cancer. So this process have been used for the last 25 years very nicely in this facility. The promise of this treatment is, as you can see in this graph, this is a graph of 3 donors blood glucose profile for 24 hours, and here you can see that just how irregular it is, and there is no control for blood sugars. But more importantly, the red circle is the dangerous lows, which means in the middle of the night the blood glucose of the patient goes below 3 millimolar or so they can go under glycopenic shock and sudden death and maybe in coma. So many diabetic patients, they died in bed during the midnight because of hypoglycemia. But after transplantation, these 3 patients have regained the very robust and rock-steady control of blood sugars, as you can see in this graph, clearly stating the promise of transplantation approach. And here is a graph that you see that when we do this for almost 25, follow-up of 20 years or so, 75% of the patient are insulin-free right after the 1 year of transplant, and they remain insulin-free for a long time. But majority of the patient have start declining the function over time by 3 years and 5 years. And this is because of the heavy doses of immunosuppression drugs, which has a negative impact on the transplanted cells. So the patient again become diabetic after a prolonged period of time. And so they may have to go for top-up dose, for second dose or third dose of transplant in order to regain the function again. But this is challenging, and that's why islet transplantation is not widely acceptable for majority of the donors, because we don't have so many organs to transplant every single diabetic patient in this world. So what are the alternative strategies to replenish these beta cells? I for almost 20 years in this field, identifying and exploring the answer for this question. And so I studied several cell types, including bone marrow stem cells, adult stem cells, embryonic stem cell-derived islet regeneration, autologous or allogenic cell therapy. But basically, the answer is like how we can really generate beta cells from these stem cell sources. So there are 2 ways we can get this. Beta cell replication, which means existing beta cell divided into 2 and 4 and 6 and so on. And so basically you can increase beta cell mass, or we can get a beta cell differentiation where endocrine progenitor cells they can actually differentiate into new beta cells. That's called beta cell neogenesis. Um, when I was a PhD student, I focused generating new islet cells from multiple stem cell sources, and my PhD was confined to utilizing sources from skin fibroblasts, bone marrow, stromal cells, or pancreatic stem cells, and basically use growth factors such as one of the growth factor I discovered was a sortisin small molecule, an activin A, that was very helpful in developing and differentiating these stem cells directly into stem cell islet cells. And we also identified the mechanism of differentiation, which was published in these two papers. In the second study, what we did is we wanted to know how these stem cells are basically differentiating into islet cells, and so we labeled these stem cells with GFP protein, exogenously, and then transplanted the labeled stem cells with GFP into into the tailvein of this mice, so that they can go and reach to the damaged pancreas of this mice. And then we, in parallel, we also treated these mice with growth factors such as activin A. What does it allows us basically, it forces these labeled stem cells to go into the pancreas and the rest over there, and all started differentiating into new beta cells. So if that happens, the new beta cells have started expressing insulin as well as GFP. That allows us to really monitor that these cells have become true newly regenerated or differentiated stem cells. So these results are that's what this paper talks about, which I published in during my PhD. So I continued the study when I came back to Canada in 2013, and during my postdoc studies, then that's where I started learning this mechanism more deeply. And what we did over here is basically I did the similar study where for lineage tracing of stem cells in mice. So here you can see the mice which has a progenitor marker, which is called mutagenin 3 or NGN3, which is a label for the progenitor marker that gives rise to pancreatic stem cells and essentially develop into beta cells. And on a reporter mice, which is called a GFP mice or Z is green, when you crossbreed these two mice, you can get a double knock-in mice with neogenin 3 and Z green. And idea is basically, if somehow we can stimulate the resident stem cells in these mice with any growth factors such as GLP-1 in this case, then the GLP-1 will trigger the expression of green protein in some of the cells in the pancreatic ducts where the neogen 3 is expressed. And if the neogen 3 expressed, that labels the cells with GFP protein permanently. And this can be traced down to all the way to the beta cells when they start developing and differentiating from the progenitor cells. So if the new beta cells produce insulin and they also have the green protein, that means that new beta cells are coming from differentiation. And this is the picture that you see over here that clearly shows that the red color is completely overlapping with the green color, indicating a yellow color. That means that these beta-like cells are differentiated from progenitor cells. This study was published. And then later in my second part, I tried to explore more into the mechanism, where I found that the mechanism of this function is associated by expression of one of the protein kinase. And found these particularly to diabetes patient, so highly expressed compared to all different members. And how to really prove this is we took a islet health donor and diabetes and with diabetes, and basically
फिर से
they,
वो प्रॉब्लम आने लगा है बीच में गायब होने का.
okay.
हाँ बीच में ठीक हो गया था. Okay, please continue.
Okay, right. What is we take these islet cells, and when we knocked out and overexpress lin in these beta cells. So when we overexpress lin in these beta cells, we found that there is a higher insulin secretion, higher beta cell proliferation, and low cell death. Versus when we knock out the lin completely in these beta cells, then we will have low secretion, low proliferation, and higher cell death. That completely indicates that lin expression is very, very important for beta cell development and differentiation. So that was my postdoc study, and this is in clinical trial now. Then I actually come to more translational space, and I joined James's lab over here. And this is the lab which I developed since 2018 to over here. This is a brand new lab which is focused on cell therapy manufacturing, and I lead the team, the process development team and the clinical team over here. We use these vessels or bioreactors to manufacture stem cells and islets, which I'm going to describe here. So what is the idea? How to cure diabetes with pluripotent stem cells? The idea is, if we have a diabetic patient, let's say, and if we get a blood sample from this patient, make a reprogram an IPS line by using Sendai viruses. So once we get an IPS line and stable clone, we can be able to differentiate islet-like cells that can produce insulin and regular glucose. And if we can get that, then we can transplant these cells into the same patient from which the blood was taken in an autologous manner. So that means we can have an unlimited supply. This recipient doesn't have to depend on the donor because it's the same cell, so they can get their own cells. Because it's their own cells, so body will not reject, and the immune system of the same person will not reject these cells, so they do not require immunosuppression or powerful anti-rejection drugs. And this will eventually reduce the risk of infections and growth versus graft versus host disease and cancers and mutation. So it has a really strong promise to treat diabetes in these patients. So this is basically the new phase of modern medicine, where the cells are basically working as a living drugs, and you don't require any drugs or insulin or anything. And if we can do this right, so this cartoon shows how we do that. We take blood, we make stem cells, we bank stem cells, and then we can expand and manufacture stem cells in large capacity. Then we differentiate stem cells into islet cells, and then put them into automation and robotic machineries to manufacture into very massive and large scale. And then finally, it can be transplanted to the same patient. And this cartoon looks very simple, but it's not very, very simple. It has a lot of challenges, particularly. And that's what we are trying to publish and work around on this idea. So theoretically, it is possible that from pluripotent stem cells, we can make islet cells in a 6 to 7 stage of differentiation process. And once we get these islet cells, which are producing insulin and green cells over here, then you can transplant these into the mice, and within 3 to 4 months of transplantation, these cells were able to regulate blood sugars in these mice. Several people have actually developed and adopted this technology and tried manufacturing islet cells successfully in plates and small dishes and well plates, 6 well plates or 24 well plates, and so on. However, the process is very limited, the yield is very low, and it requires pooling with multiple vessels, so that creates a lot of heterogeneity in a final cell product. So what we have a solution over here is basically we are using a bioreactors instead, which means it's like a specialized bottles which has a media filter in the cells, and the cells are just growing in 3D suspension. And I will show you in a video in the next slide how that works. But we're basically using these bioreactors to replicate all of these stages that can be replicated into and differentiate into mature beta cells. So the benefit is, like, we can have a very high yield. We don't require cooling from multiple vessels, and we can make islets without manipulation. And this is the reason why, because the cell therapy manufacturing companies are basically focused more into 3D culture system. The benefit is because they have a very strong promise for scale-up capacity. And so there is a very strong market in the US as well as in India, particularly for 3D culture system. And many companies in globally, including Asia Pacific and India, There's 20% of these companies are really gaining a market share for 3D suspension culture system for cell therapy manufacturing. So just to give an idea, what does that mean? This is a picture. In order to make a cell, if I have to go into plates, I have to make about 200 to 400 plates just to give like 20 to 50 million cells. This can be replaced by stacks of plates like that, which are more automated culture system available now. But they can also make only, 100 million cells or maximum 200 million cells in a scale-up process. What we are trying to bring a new technology over here is using many bioreactors, which could be from zero point 0.1 to zero point 0.5 to 3-liter capacity. And the beauty of this technology is we can easily generate over 100 million cells to 3 billion cells, 3 into 10 to the power 9 cells in a 3-liter capacity. These cells are enough for transplanting one patient of diabetes particularly. So that means that for a single bottle can actually give us enough cells that is required for a cell transplantation in one patient without pulling hundreds of tubes and vessels, compared to these areas. So we published a paper recently, a year ago, where we have shown that how we can use these vessels for generating stem cells in these bottles. And basically, the cells can be transferred from plates to these reactors, either in 0.1 liter or 0.5 liter. And when we do that, we basically get clusters of cells or 3D aggregates. So that's how the cells go in 3D suspension and 3D condition. We found that these cells are highly pluripotent, as they can maintain the expression for pluripotent markers, as shown over here in flow cytometry, compared to the plates. So these cells are obviously very highly pluripotent. They also maintain their phenotype and morphology, so there is no change in cell shape and size and viability when you culture them into suspension reactors. This is a picture that you see. With flow cytometry, they are very much pluripotent, as they're expressing pluripotent markers, and confirmed by immunochemistry. Every single cell over here is expressing pluripotent stem cell marker. So we're now replicating this and then basically transforming to a 3-liter expansion capacity, so we can really massively expand the process. And not only the expansion, we can also differentiate the islet cells from these bioreactors. And here it is an example. We can get the stem cells that we have expanded. We can quickly transfer these cells into differentiation into 7 stages, as you can see over here. So basically, we don't have to manipulate or break the process. We can continuously go from expansion to differentiation. The whole process can take about 2 to 3 months. But in the end, you can see that how these islet-like clusters can be generated from multiple iPSC donor lines in both either 0.1 liter capacity or 0.5 liter capacity. And then these cells clearly show the presence of insulin, as you can see over here in green, and glucagon in red, indicating that these cells are very hormonal positive and they have the capacity to regulate blood glucose. We can get easily over 200 to 400 million cells from a small reactor. This shows a very strong promise for scalability in terms of doing a clinical trial in patients. So we know that we can generate the islets. We know that we can manufacture them, but they're also very enriched for genomic markers that shows the identity into pancreatic lineage compared to the cells that has not suspension. And when we transplant these cells in mice, in diabetic mice, they rapidly able to reverse diabetes in mice, as you can see over here in dark brown line, compared to the faded line, which is like an conventional protocol without 3D. So that clearly indicates that the 3D suspension differentiated grown cells have the capacity to rapidly reverse diabetes within 30 days, and they can maintain the glycemia in these mice for a prolonged period of time. This is the graph histology, where you can see the green cells are insulin and the red cells are glucagon, indicated that the transplanted cells survived developed and mature in these diabetic animals post-transplantation. So what is the future of this medicine? We can make stem cell utility for manufacturing cell product. We know that stem cells has the promise, but in future, the focus on regenerative medicine is more for the diagnostic, improving the diagnostics, the medical imaging, how these cells grow inside in the body, how these cells are able to develop and differentiate into multiple cell types. We can use nanotherapeutics where we can use targeted drug delivery or and tackle autoimmunity or immune system to use such as monoclonal antibodies along with the cell types to regulate autoimmunity and type 1 diabetes. We can also use these cells for vaccine production or maybe a regenerative medicine formulations for biomaterials and encapsulation devices. So the one of the example that I would like to show you over here, the technological advancement is so important that integrated use of artificial intelligence or AI and machine learning is really critical for modern medicine development. And this is one of the study we did and published last year. Where we show that how we can use AI to predict the iPSC reprogramming success and really if the cells are reprogrammed into pluripotent stem cells or not from patient and diabetic patient stem cells. So what we did basically, we take an iPS cell, we did a time-lapse imaging for several days, generated more than 30 to 40,000 image data set, and then integrated the deep learning and AI module into this. And this is how you see when the stem cells grows, they actually expand and grow in time. So the microscope captures the live cell imaging in the real time. And then when we throw AI modules, the AI was able to detect something which a normal human eye cannot detect. So we using the machine learning algorithm, we were able to predict that the cells that are going to be a true stem cells in future compared to the cells that are that may fail in future. They might be looking time, but eventually these cells gonna fail in future and will become more differentiated cells compared to the iPS cells that are shown over here green. And this is how AI could separate out, and this will allow us an ability to selectively enrich and predict and pick these cells as true stem cells and not the. So the power of AI is immense over here that allows us to really control the cell therapy product. And this is another example. What we're trying to build over here is the use of automation and robotics, precision medicine cell therapy manufacturing. There are robotics and automation has gone far beyond the technology where they can simply replace humans and the robots can actually do the entire cell processing, manufacturing, cell production, selection, identification, and characterization of iPSC clones. And basically the essence of this is if the robots can actually do this, we can remove the heterogeneity, the variability, man-to-man variability during the manufacturing process, because humans are so vulnerable. Like technician who is doing the work every single time, they can incorporate errors, but the robots can actually do the work so precisely that every time we're going to manufacture the cell product for one patient to second patient to third patient, is going to be same every single time. So there will be no variability. And this is why I think robotics will come up as a future of cell manufacturing. This
How
is
many
just one
slides left?
example. So I think I'm pretty much done now.
Okay, please.
Yeah. So we have this clinical trial thing going on right now, and we're working for the clinical trial, working with regulatory agencies, and we are going to do the clinical trial in type 1 diabetic patients and total pancreatectomy patients and 1 year follow-up. This is a GMP facility that we already have in-house and we're going to produce. That's the clinical trial team, including James over here. He's the director of the program. Myself, I'm manufacturing, and the other partners who will be doing the post-transplant care for this trial. So in summary, I told you about the stem cells as really a next generation therapy to treat diabetes. It allows a precision medicine and tailored cell therapy production. Robotics and automation can really be integrated to make more technological advancement and probably eliminate the need for drug and replace organs and restore function. So with this, I would like to thank you for your attention.